Hi everyone, today we are learning the difference between gram-positive and gram-negative bacteria and how to identify them using the gram staining procedure. Let's begin with the basic difference between gram-positive and gram-negative bacteria. The gram-positive bacteria have a thick peptidoglycan cell wall. Because of this structure, they retain the crystal violet stain during the gram staining process, and under the microscope they will appear purple. In contrast, the gram-negative bacteria have a thin peptidoglycan layer and an outer membrane rich in lipopolysaccharide. These bacteria lose the crystal violet during decolorization but take up the safranin counter stain, so they appear pink or red under the microscope. The gram staining helps us to differentiate bacteria into gram-positive and gram-negative based on their cell wall structure. This makes it easier to identify the type of bacteria present in a specimen. The gram staining steps. First, crystal violet. It is a primary stain, and this dye stain all bacteria purple during the initial step. The second, that is iodine, the mordant. What iodine will do? The iodine fixes the crystal violet dye to the peptidoglycan layer, forming a stable diiodine complex. And the third component is alcohol or acetone, the decolorizer. This step removes the purple stain from the gram-negative bacteria while the gram-positive bacteria retain it. Finally, safranin, the counter stain. The safranin stains the decolorized gram-negative bacteria pink or red, while the gram-positive bacteria stay purple with the crystal violet. Together, these steps allow us to clearly distinguish gram-positive from gram-negative bacteria under the microscope. First, we need to heat fix the slides. Using forceps, gently pass the slide quickly over the flame several times. The key is to heat it lightly. Don't overheat or burn the specimen. Once fixed, place the slide on the staining tray. Flood the smear with the crystal violet and allow it to sit for 1 minute. Crystal violet will stain all the bacteria purple initially, then rinse the slide with distilled or tap water. Next, add iodine solution. It is the mordant. Let it remain in the slide for 1 minute, and the iodine bind the crystal violet and form a complex that helps the stain stay attached. Rinse again with water. Now tilt the slide slightly, gently flood it with a decolorizer, the acetone or alcohol for 1 to 5 seconds. These steps remove the stain from the gram-negative bacteria while the gram-positive bacteria retain the purple color. Immediately rinse it with a distilled or tap water to stop the decolorization process. And the last step is apply safranin for 30 seconds. The safranin will stain the gram-negative bacteria pink, and while the gram-positive bacteria remain purple. Rinse with a distilled or tap water. Finally, dry the slide gently using an absorbent paper and place it upright to finish drying. Under the microscope, you will now be able to distinguish gram-positive bacteria that is purple in color and the gram-negative bacteria that is pink or red in color. This simple staining technique reveals the microbial population present in the samples. If you find this tutorial helpful, please like, share and subscribe for more microbiology and veterinary pathology related videos. Thank you.